Now I am going to teach sexual reproduction in flowering plants. Sexual reproduction. There are various events are possible in sexual reproduction. Here, first we have to understand the structure of flower. Before going to the flower, we have to understand what is called pre-fertilization events. So the pre-fertilization, we can say inflorescence formation, floral buds and then only flowers. But without these kind of informations, we study in inflorescence and everything in the structures of plants, that we will morphology and anatomy. Now we are going to see what is called structure of flower very clearly. In flower, we got a androecium, what we call as a androecium and the gynoecium. Two structures are there. The two main structures, androecium and gynoecium. The androecium contain important thing called stamen, and stamen got two structures. One is filament and anther. This filament and anther is a very important structure for the male part, androecium. The filament normally we can say its proximal end is attached with the thalamus or petals of the flower. It is generally long and slender stalk. Its proximal end is attached to the thalamus or petals of the flower. The anther, that particularly the terminal one, typically bilobed structure, and each lobe has two theca, so they are diathecious. So usually the anther is actually a bilobed structure that has normally two theca, so we call them as each lobe has two theca, we call them as a diathecious. And a transverse section of anther, if you study the transverse section of anther, the anther is a tetragonal structure. Anther is a tetragonal structure. Anther is a bilobed structure. Filament is a long and slender part. Its terminal end bears with anther, it's a bilobed structure. Each lobe has two theca. So each lobe has two theca. If you say two theca is there, it can be explained as what we say. So this particular thing, two theca is there. So the two theca is a characteristic feature, what we call it as diathecious. So the transverse section of anther, if you go for, it is tetragonal in nature. It is tetragonal in nature. And this particular structure consists of 4 microsporangia. This tetragonal structure contains 4 microsporangia. And microsporangia This is a location where a pollen sac, microsporangia, develop further and they become a pollen sac. And this pollen sac, they contain or packed with pollen grains. So let me discuss the microsporangia, the four microsporangia, which is going to give pollen sac very clearly. So microsporangia. Talking about microsporangia in detail. Specifically, it is surrounded by four-walled layer: epidermis, endothecium or endothecium, middle layer, tapetum. Microsporangia is covered by or made by a four layer. Nearly they are circular in outline. When you see it is circular in outline, that is made up of epidermis, endothecium, middle layer and tapetum. So remember, the outer three layer performs a function of protection. What are they? These three layers perform the function, specifically protective function. Tapetum nourishes the developing pollen grains. So this tapetum plays a vital role. The tapetum possesses a dense cytoplasm and generally have more than one nucleus. So they got dense cytoplasm plus more than one nucleus. It's a function of tapetum. The other one is when we enter the next point of this concept, microsporogenesis. That means the formation of microspore, that is pollen grains. The anther develop a sporogenous tissue, undergo meiotic division to produce microspore tetrads. So this anther, they got a microsporangia that got a sporogenous tissue, that got a what we call as sporogenous tissue. So this sporogenous tissue, what they will do, they produce an important thing called microspore tetrad, and each one is a potentially pollen grain, or when you say microspore mother cell. Each one is a potential microspore mother cell. So the microspore tetrad, I already told you, this is actually tetrad. Out of that, each one is a microspore mother cell. Microspore mother cell. This microspore mother cell, they normally leads to the formation of microspore through a process called meiosis. Through a process called meiosis, they form microspore. I repeat, during the microsporogenesis, sporogenous tissue involved in formation of microspore tetrad, they got microspore mother cell that able to produce microspore. Very simply speaking, this information. And coming to the next point, the pollen grain is ready right now. Inside each microsporangium, thousands of pollen grains are formed. Each and every microsporangium, there will be a sporogenous tissue. They produce microspore tetrad. Microspore tetrad produce microspore mother cell. That microspore mother cell will produce microspore. You can expect around in each and every microsporangium around thousands of pollen grains. You can expect. So let me talk about pollen grains in detail. If you go for the pollen grains alone, the pollen grain has two-layered concept. Pollen grain is a two-layered concept. When you go for the pollen grain, the outer layer is exine, inner layer is intine. Exine, intine. Exine is a hard outer layer which is made up of sporopollenin. Sporopollenin. This sporopollenin is a which give you a toughest nature which cannot be digested by any enzymes. Very strong in nature. And coming to the next category which is intine. This intine is an inner wall which is made up of cellulose and pectin. Intine is made up of cellulose and pectin. Exine got a small pores where the sporopollenin is absent. So we can say it is discontinuous. This is continuous. The difference between exine and intine is actually discontinuous and continuous. The pollen grain consists of two types of cell. The two types of cell, one is vegetative cell, another one is generative cell. One is these two cells, one is a large and irregularly shaped nucleus, and it normally has abundant food reserve, that is vegetative. Generative is a small and normally it is spindle shaped with a dense cytoplasm and a nucleus. So there are two types of cells, one is a large and one is small and spindle shaped. All those things are very clear. And coming to the point, this particular formation, if you go for 60% of angiosperms, their pollen grain shed at two-celled stage. In another, where we can say the generative cells divide mitotically to give rise to two male gametes before pollen grains are shed. That means we can say three-cell stage. So we can say the pollen grains are divided in two-cell stage, or maybe pollen grains are shed at the three-cell stage. In two different conditions, this pollen grains may shed. I repeat, 60% of angiosperms, pollen grains are shed at two-cell stage. And in other generative cells, in others, the generative cells divide mitotically to give rise to two male gametes. So this, after production of two male gametes, it becomes three-cell stage. So after this, vegetative cells and generative cells, okay, they shed at three-cell stage. So possibility, this will be divided into other two cells and then it becomes three-cell. They may divide in a three-cell stage. Once they shed, the pollen grains have to land on stigma. So pollen grains specifically, remain viable, and it is depend on prevailing temperature and humidity. So what we have to understand is, actually, some pollen grains got the viability of 30 minutes. Example, rice, wheat, etc. Some members, normally we can say Leguminosae, Rosaceae, Solanaceae have viability more than a month. Pollen grains, some plants have what we say leads to allergic, we can say parthenium or carrot, grass, or allergic for some people. It leads to a chronic respiratory disorder like asthma, bronchitis, etc. Pollen grains are rich in nutrients, so some tablets are developed based on these pollen grains. And we can store the pollen grains in liquid nitrogen around minus 96 degrees Celsius. You can keep it. The next one is the pollen grains. About pollen grains, I just repeat, it is from the anther. The pollen grains can be prepared from the microsporangia, sporogenous tissue, prepare it, produce microspores. Okay, so microspores are nothing but pollen grains. In gynoecium we got a stigma, style, ovary, megasporogenesis. That's we are going to discuss about the spore formation. Style, stigma, ovary. Stigma, style, ovary. This ovary is a bulged part. Ovary is got an ovarian cavity called locule, in which placenta is located here. Placenta is located, and the placenta, maybe we can say arising from the placenta are the ovules from the placenta. So this is, if you say this is ovary, from these ovules arise from the placenta. So number of ovules in an ovary maybe what we say vary. Maybe one in wheat, paddy, mango, etc. Many in papaya, watermelon, orchids, etc. And when you come to the point of production of ovule from the ovary, it is a small structure attached to the placenta by means of a stalk called funicle. This got attached with this with the funicle. The junction where the body of the ovule and funicle fuse is called hilum. The ovule has one or two protective envelope called integuments. Previously we discussed the protective covering of the pollen grains as exine and intine. Here it is integuments. In ovary we discussed that 4 different layers got, and that tapetum is going to prepare, nourish microsporangium, sporogenesis tissue, and then we got microspores like that. Here where we can say it is integuments. The ovule has 1 or 2 protective envelope called integuments, and there will be a small opening what we call as a micropyle. Small opening will be a micropyle. Opposite to the micropyle end is a chalasa, a basal part of ovule. So ovule got opening micropyle. The opposite region we can call as a chalasa. Micropyle opposite is called as a chalasa. And enclosed with the integuments, there is a mass of cell called nucellus. It contains reserve food material. It is not nucleus, it is nucellus. It contains reserve food material. Located in the nucellus is the embryo sac. It's a female gametophyte. How we explain that the male gametophyte. The same way here, the female gametophyte. And ovule is generally as a single embryo sac formed from a megaspore through meiosis. So let me discuss about the megasporogenesis, megaspore ovule formation. The formation of megaspores. Ovule generally differentiated into megaspore, mother cell in micropolar region of the nucleus or nucleus. It is large cell containing dense cytoplasm and a prominent nucleus. The megaspore mother cell undergo meiotic division. It results in the production of 4 megaspore. So during megaspore formation, megaspore mother cell undergo meiotic division and results in the formation of 4 megaspore. Female gametophyte, the embryo sac. I already told you it is placed in the nucleus region, and ovule generally has only one embryo sac. That embryo sac is in a majority flowering plant, one of the megaspore is functional while others 3 are degenerate. If even they produce 4 spore, one is functional, other 3 are degenerate. The functional megaspore develop into a female gametophyte. The method of embryo, that is what we say, the embryo sac formation from a single megaspore is termed as monosporic development. What we say, the method of embryo sac formation from the single megaspore. Just now I told you how the megaspore will be formed. This megaspore will be formed from megaspore mother cell, as previously how we discussed microspore mother cell, how microspore got generated. Here also from megaspore mother cell, megaspore will be produced. 4 out of this, one is functional, other 3 are not functional. The functional one is going to form that only one thing, that is embryo sac formation from single megaspore, what we call it as monosporic. That one we can call as a female gametophyte or female gametophyte, that is called embryo sac. And this female gametophyte embryo sac formation took place. The nucleus of the functional megaspore, how that particular nucleus, that embryo sac is forming, that we have to explain it very clearly. The nucleus of the functional megaspore divide mitotically to give 2 nuclei and which move to the opposite pole and forming 2 nucleate embryo sac. The 2 more sequential mitotic nucleate division results in the formation of 4 nucleate and later 8 nucleate stages of embryo sac. So 2 nucleate embryo sac, 4 nucleate embryo sac, 8 nucleate embryo sac. All those things normally we can say happening in the nucleus of the functional megaspore. Okay, so the functional megaspore divide mitotically. They give 2 stage, 4 stage, 8 stage embryo sac. These divisions are strictly free nuclear, or we can say nuclear divisions are not followed immediately by cell wall formation. After the 8 cell stage, cell walls are laid down, leading to the organization of typical female gametophyte or embryo sac. 6 to 8 cell nuclei are surrounded by cell wall and organized into cell. The remaining two nuclei, polar nuclei, and are situated below the egg apparatus in the central cell. So once again repeating the point, the functional megaspore divide mitotically to form two cell or two nuclei stage is called two nucleate embryo sac. Later sequentially mitotically nuclear divisions results in the formation of 4 nucleate, later 8 nucleate. The divisions are, very simply speaking, nuclear divisions are not followed immediately by cell wall formation. After 8 nucleate cell walls are laid down, leading to the organization of typical female gametophyte or embryo sac. This is surrounded by cell wall and organized into cells. The remaining two polar nuclei are situated below the egg apparatus in the central cell. So how that is distributed, the nucleus is distributed in the distribution of cells within the embryo sac. We are discussing about different stages, right? So that how the distribution takes place. 3 cells are grouped together in the micropolar region, that means the opening region, and constitute the egg apparatus. The egg apparatus consists of 2 synergids and 1 egg cell. 3 cells, out of that 2 synergids and 1 egg cell. The synergids have a special cellular thickening at the micropolar region tip called the filiform apparatus. It helps to guide the pollen tip into the synergid. 3 cells are at the chaliceal end are called as an antipodal. The large cells has 2 polar nuclei. The typical mature angiosperm embryo sac is 8 nucleated 7 cell stage. So that's all about the embryo sac description. So repeat, the cells distributed like 3 cells and then 3 cells at the end of micropolar region and the other 3 cells in the chaliceal region. The chaliceal region is, and this and that together is actually 6 cell, the middle is 2 cell. So these are all basic structural information about the, what do you say, megaspore. So when we talk about pollination, I told you the pollination is possibly by autogamy, gynogamy, xenogamy. So these are the way. Autogamy, transfer from anther to stigma of the same flower. That is called as an autogamy. So this autogamy and gynogamy and xenogamy. Here the pollen grains transfer from the anther to the stigma of the another flower. Xenogamy, the pollen grains from the anther to the stigma of the different plants. So that's all about this topic today.